Good day, ma'am. Our lesson entitled Midterm Laboratory Exercise, Semen Analysis. Before we proceed to our lesson, let's start with the lesson objectives. Students should be able to explain the importance of semen analysis and analyze the forensic examination of semen. And second, the contents. First slide, title, objectives, slide contents, and presenter's identity. Second slide, description of semen, sperms, and semen analysis. Third slide, difference between presumptive test for semen and confirmatory test for semen. Fourth slide, steps in collecting DNA evidence. And last slide, importance of semen analysis in criminal investigation. Now I will discuss the semen, sperms, and semen analysis. Let's start with semen. It is the fluid released through the penis during orgasm. Semen made up of a fluid and sperms. The fluids come from prostate, seminal vesicle, and other sex glands. Next is the sperms. It is the male reproductive cell or gamete in anisogamous forms of sexual reproduction. And last, semen analysis. It is the great value in a medical legal practice like alleged rape cases or sexual assaults. The primary aim of forensic semen analysis is to sample and examine smears or other biological material take from the assailants, the victim, or stains found in the clothes and other evidences in the crime scene. There are three presumptive tests for semen. First, visual and alternate light source. Semen can be detected with the naked eye in clothing and undergarments or through the use of alternate light source such as ultraviolet light. And second, acid phosphatase test using standard chemical reaction laboratories and analyze a given stain for the presence of these enzymes. In the presence of certain stain, alternate phosphatase will produce a dark purple color in less than a minute. And last, the prostate specific antigen. It is also known as PSA or P30, and it can be detected in semen. PSA is produced in high amounts by the male prostate glands but can also be found in very small amounts in fecal matter and breast milk. Next is the confirmatory test for semen. There are two confirmatory tests for semen. First, Christmas tree stain. It is a positive identification of sperm cell. The first stains the neck and the tail of the sperms in green and blue, whereas the latter one stain the sperm head and acrosomal cap with the red and pink. Next is the RSID semen strip test. This test provides sensitivity and specificity to human semen. Similar to the pregnancy test strip, RSID semen test identifies the presence of the seminal vesicle specific antigen. Now, steps in collecting DNA evidence. First step, absorb suspected liquid semen on a clean cotton swab. Air dry the swab and pack in a clean envelope with sealed corners. Second step, submit small and dry suspected semen stained to the laboratory. Pack in clean envelope with sealed corner to prevent stain removal. Third step, documenting the scene. Develop written documentation, photograph, locate, and interview victims and witnesses. Fourth step, processing evidence at the scene. Organize evidence, processing and control scene contamination. And the last step, completing and recording the scene. Make sure all the investigative steps are documented and release the scene and submit the report. And for the last slides, importance of semen analysis in criminal investigation. Semen analysis is very important in assault cases as it is very useful for detection of semen in clothes, isolation and identification of seminal stain found on various suspected such as clothes, bedsheet, pillows, blanket, and etc. And analysis of various um, protein present in semen or seminal stain. Detection of sperm cell in the vagina and other body part and fecal matter examination. And that is the importance of semen analysis in criminal investigation. And that would be all for this midterm laboratory exercise for semen analysis.